Hello, my dear students. Did you know that some plants can grow new individuals from just a leaf or stem? Or that certain tiny animals can reproduce without needing a mate? Today, we are stepping into the fascinating world of asexual reproduction, a mode of reproduction where only one parent is enough to produce offspring. There is no need for male or female gametes, and yet new life begins. You will learn how simple organisms like amoeba, hydra, and planaria, as well as plants like potato, bryophyllum, and ginger, multiply through methods such as binary fission, budding, spore formation, and vegetative propagation. It's nature's clever way of ensuring life goes on, even without seeds or fertilization. Let's get started and explore how a single life from can create many more. Reproduction is fundamentally the biological process through which living organisms produce new individuals of their own kind, ensuring the continuity of the species. Types of reproduction. Reproduction basically have two types: asexual reproduction and sexual reproduction. So asexual reproduction involves a single parent, no involvement of gamete or sex cells. The key feature is offspring are genetically identical to the parent in case of asexual reproduction. Examples: binary fission in amoeba. The amoeba, this is the diagram showing the amoeba splits into two daughter cells, each genetically identical to the parent. Second one is the budding in yeast. This is the diagram showing budding in yeast. A small bud forms on the parent body. It grows and then detaches as a new organism. In case of plants, asexual reproduction can occur via vegetative parts like roots, stem, and leaves. Now we'll study the sexual reproduction. It involves the fusion of male and female gametes, that is sperm and ovum, which is called as fertilization, and then zygote formation occurs, and which is followed by the embryonic development. So fertilization is the male and female gametes unite, zygote the fertilized egg cell, embryo the developing organism after cell divisions. Fertilization can be of two types: external fertilization and internal fertilization. When fertilization occurs outside the body, like fishes, frogs, etc., they are called as external fertilization. And when fertilization occurs inside the body, then in case of birds and mammals, these are called as internal fertilization. Gamete producing organs may be of two types: male gamete or female gamete. Testes produce sperm, which is a male gamete, and ovaries produce ovum or egg, which is female gamete. Some animals, snails, tapeworms, have both organs like hermaphrodites. In plants, unisexual flowers has either male or female reproductive parts. Bisexual flower has both male and female reproductive parts. Now we will study asexual reproduction in plants and animals. Asexual reproduction is basically the mode of reproduction where a single organism produces offspring without the involvement of gametes or sex cells or fertilization. Characteristic features of asexual reproduction: it is the first process, it ensures quick multiplication, single parent involved, no male or female gametes are needed, offspring are identical, the new individuals are clones of their parent. Types of asexual reproduction: binary fission, multiple fission, regeneration or fragmentation, spore formation, budding, vegetative propagation in plants. This is the image showing budding in hydra. This is the another type of asexual reproduction called as parthenogenesis. This is fragmentation found in planaria, and this is the binary fission found in amoeba. Now we'll study binary fission. Binary fission is common in unicellular organisms like amoeba, euglena, chlamydomonas, sea anemone, etc. It occurs when food is abundant and conditions are optimal. This is the image showing binary fission, and this is showing the types of binary fission found in amoeba, euglena, paramecium, and serratium. In case of binary fission, nucleus divides into two. Constriction appears in the cytoplasm. This is the constriction. The cell splits into two daughter cells, each with one nucleus, and then daughter cells grow into new individuals. Daughter cell থেকে নতুন individual তৈরি হয়. Multiple fission. It is found in organisms like Plasmodium, which is a malarial parasite. It happens under harsh conditions like extreme temperature, scarcity of food, etc. Parent forms a cyst which have a protective wall. Inside the nucleus, it divides repeatedly, and each nucleus is surrounded by cytoplasm to form daughter cells. On favorable conditions, the cyst bursts and multiply daughter cell, and multiple daughter cells escape. There is a fragmentation and regeneration. If an organism is broken into fragments or parts and each piece contains a nucleus, it can grow into a new individual. This is the example, this is the image showing the fragmentation found in planaria. Organisms broken into several parts and each piece will contains a nucleus and it can grow a new individual. And the regeneration, if an organism loses a body part like hydra, it can regrow. However, a fragment without nucleus can regenerate and dies. What is the spore formation? It is found in hydra, fungi and some algae. The sporangium is a sac-like structure found at the tip of hyphae that produces numerous spores. Spores are released into the air or land in a suitable conditions and germinate into new organisms. Example, the bread mold, which is Rhizopus, Mucor, Chlamydomonas, etc. Now let's study budding. Budding, a new organism develops from a bud or small outgrowth on the parent's body. Just like budding in yeast, found in case of unicellular organism, a bud forms on one side. The nucleus divides and one daughter nucleus moves into the bud. The bud grows and detaches to become an independent cell. This is the image showing the budding in yeast. And budding in hydra, which is a multicellular organism, a small bulge or protuberance or bud forms. The bud grows and develops tentacles. It later detaches and becomes a new hydra. This is the fragmentation, regeneration, spore formation, and budding process. Their key features and their examples are shown in the table. Vegetative propagation in plants. Vegetative propagation is a form of asexual reproduction in which buds present on vegetative parts like roots, stems, or leaves grow into a new plant without seeds. It can occur naturally or through artificial methods. Natural vegetative propagation or role of roots. Roots not only absorb water and minerals from soil but also act as storage organs and support vegetative reproduction through buds. This is an image showing the type of vegetative reproduction which is naturally occurring. This is another type of vegetative reproduction. Perennation, the ability of plants to survive from one generation to the next even in unfavorable condition like drought, winter, etc. In this case, roots like tuber, rhizomes and corms store food and have buds that help in both, which is the survival, that is perennation, and reproduction is the vegetative propagation. This is the table showing different types of vegetative propagation found in taproot, tuber, fibrous root, or other examples like mint, guava, and they use the type of root like true root, adventitious root, and propagate by their adventitious bud or buds on fibrous root or buds from new plants. Now let's study roles of stems in vegetative propagation. Stems. Underground stems store food, remain dormant, and help in both. Perennation, it's surviving adverse seasons like cold, dry, etc. Perennation giving rise to new plants in the next growing season. Through these underground parts resemble root, they are actually stems because they possess nodes, internodes, buds, and scale leaves. These are showing the plant stem which undergo vegetative propagation. Rhizome of ginger, a horizontal underground stem, bears nodes, buds, adventitious roots, stores food like starch and proteins. Its shoots arise from the bud. Note the rhizomes help both in storage and reproduction. This is the rhizome of ginger, and this is the growing plant from the ginger or rhizome of ginger. This is potato tuber. This is potato tuber. Is swollen underground stem, not root. Eyes or lateral buds. We have the image showing the eyes or lateral buds develop in shoots forming new plants. Storage stores a large amount of starch. The eye is a bud. It's not just a spot. This is a potato plant. Bulb of onion, a short underground stem surrounded by fleshy scales, leaves storing food. It has terminal bud, axillary bud, fleshy buds, basal stems or disc. Note replanting bulbs lead to development of new onion plants. This is the image showing the onions. This is the onion showing the bud, roots and scales. Subaerial stems, horizontal stems that grow along or just under the soil surface, function it don't store food. They are purely for propagation. After rooting at nodes, each runner gives rise to a fully independent daughter plant. The key features are internodes, and the elongated segments become nodes. There are internodes, and these points where adventitious roots and new shoots develop. These are nodes. Examples: strawberry and grass. Strawberry sends out runners that root at intervals to form new strawberry plants, and grass it spreads similar to form dense mat. This is the image showing the runners, strawberry and grass. Roles of leaves in vegetative propagation. Some plants form bud on their leaf margin. Example, bryophyllum. These buds develop adventitious roots when they detach soil. Process: buds or plantlets form along the leaf edges. This is the leaf edge, and their buds are emerging. When the leaf contacts moist soil, these buds send down roots, and each rooted bud grows into new plant. Example হচ্ছে bryophyllum or kalanchoe. The notches along with the leaf produces form tiny plantlets that easily root and grow when the leaf falls. Bulbils. It is a leaf-borne birds, small bulb-like structures that develop in the axil or inflorescence. These are showing the bulbil, the leaf-borne bud. It acts as a miniature bulb. Once detached, each bulbil can grow a new plant under favorable conditions. Example, agave, which produces aerial bulbils on its flower stalk. Now let's study artificial methods of vegetative propagation. When humans assist in propagation of plants using plant parts or vegetative propagules, it is called as artificial vegetative propagation. This method is used to introduce desirable traits like improved flavor, better color, greater yield, disease resistance, etc. The image showing different types of artificial method of vegetative propagation like grafting, cutting, layering, tissue culture, etc. Vegetative propagules are the part of the plant used for artificial propagation like stem, root and bud. Cutting. A stem with nodes and internodes is cut from the parent and planted in moist soil to grow into a new plant. It can be done using stem cutting, which is common, and root cutting in some plants like example rose, sugarcane, etc. These are image showing stem cutting, the cutting planted with node plant in the soil, and this is the image showing root cutting where the planted root produce new shoot. Now study grafting. A stem piece called scion from the desired variety is jointed to the root system of another plant, which is called as stalk. This is the upper part is the scion and the lower part or the root containing part is the stalk. The grafted plant combines the qualities of both scion desired fruit or flower trait and stalk produces disease resistance or strong root system. Example mango, guava, etc. Layering. A stem is bent and buried in the soil or wrapped with moist mosses in air layering. It is found in buried part, roots develop and the shoot grows into new plant. Example jasmine, tomatoes, etc. Their importance in food orchards and horticulture. This is the example of simple layering and mound or stool layering. Now let's study the important topic tissue culture or micropropagation. What is it? Tissue culture is a biological method of growing new plants from a small piece of tissue called an explant in a sterile artificial medium under controlled lab conditions. This method is also called micropropagation or cloning, as it produces genetically identical platelets from the parents. The image showing the tissue samples scraped from the parent plant and then tissue samples placed in agar growth medium containing nutrients and auxins. The samples develop into tiny plantlets and the plantlets planted into a compost. Steps in tissue culture. Firstly, explant selection. A small portion of tissue like root, shoot or meristem is cut and transferred in a nutrient medium. Example shown meristem cuttings in the sterile culture. Callus formation. The explant absorbs nutrient and becomes a mass of undifferentiated cells called callus. Callus is totipotent cell which can become any plant part. Differentiation into plantlets. Basically what happens, callus is transformed into a new medium with hormones. Cells differentiate to form roots and shoots, becoming complete plantlets, meaning the plantlets are gradually acclimatized to external conditions in a greenhouse before planted in soil. Why we use tissue culture? Grow rare or expensive plants, mass production of disease-free clones, grow plants year-round in limited space. What are the steps of plant tissue culture? A plant tissue or explant is scraped from the parent plant. The explant then placed on the growth medium supplied with nutrients and phytohormones. The explants loses specificity and divides to form a mass of undifferentiated cells called callus. Over time, small plantlets begin to develop from the callus. The plantlets are then transferred into pots or trays filled with nutrient-rich compost. What are the advantages of vegetative propagation? It is fast and reliable, no pollination or fertilization needed, works even in harsh conditions unlike seeds, preserves parental traits, offsprings or clones are they are identical in form, flavor and quality. They are useful for plants with no seeds, example banana, rose, sugarcane and orange. The seedless fruit cultivation, examples grapes, oranges. Disadvantages, it cannot produce new plant varieties naturally. Offspring can grow in dense cultures near the parent, competing for nutrients and space. There's a table showing tissue culture or micropropagation, key features, steps and benefits. Method, steps: explant, callus, plantlet and veining. Use: cloning, conservation of rare species, rapid multiplication of plants. Advantages: first, multiplication, reliable results, genetically identical, true to type plants. Disadvantages: no genetic variation, limited spread, overcrowding near parent plant. Alright dear students, that brings us to the end of today's class on asexual reproduction in plants and animals. Let us quickly recall what we learned. We understand that reproduction is the process by which living organisms produce their young ones. These are two main types of sexual and asexual reproduction. Asexual reproduction happens without gametes and includes methods like binary fission, budding, fragmentation, spore formation, regeneration and vegetative propagation. We also saw how vegetative propagation can be natural or artificial, and we discussed techniques like cutting, grafting, layering and tissue culture. These concepts are not only important for exams but also help us understand how life continues and multiplies in nature. Thank you so much for watching and learning with me. If you found the class helpful, don't forget to like, share and subscribe to support the channel. Feel free to drop your doubts or thoughts in comments below. I'll be happy to reply. Take care, stay curious, and I will see you in the next class. Biology is life. Keep exploring. Bye.